My dear students and friends, today we will start with immune system, means immunology. What is this immune system? There is a type of responses our body is having, that is effector response and the memory response. As you all know, in the current situation, the immune system of our body is very important. Functionally, an immune response can be divided into two related activities, that is recognition and response. Immune recognition is remarkable for its specificity. The immune system is able to recognize chemical differences that distinguish one foreign pathogen from our body. It means when we are exposed to some foreign particles or foreign particle in origin, that is, first we have to understand what is a antigen. Antigen, from the earlier classes we are studying, anything foreign is an antigen. No, what I am saying is anything which evokes our immune system in the body, that is an antigen. It may be protein, it may be DNA, it may be RNA, it may be pollen grains, it may be anything which evokes your immune system. That is, in our body we are having a resistance to develop disease. That is our anti-immune system. That is the effector response. What is effector response? To eliminate or neutralize the organism. In this way, the system is able to convert the initial recognition event into a variety of effector responses. And memory response, that is a very important response our body is having, because when we are exposed to some antigen and that same antigen we are exposed in the second phase, there is a primary response, and when the same antigen again exposed to that same individual, it is the tertiary response. Every time there is a response, it is very vigorous. So what is in immune system? Mainly we have to understand what is the immune system. Every vertebrates, as you all know what is a vertebrates, because we are having a vertebral column. Vertebral column means because we are having a erect posture, we are having a vertebral column. So that is those organisms which are having vertebra, those are known as vertebrates. And the vertebrates are only having the immune system, and other organisms are having some immune system like responses, but it is not immune system properly. And that is a very important thing the humans are having. Some historical perspective, the discipline of immunology grew out of the observation that individuals who had recovered from certain infectious diseases. The Latin term immunis, meaning exempt, is the source of the English word immunity, meaning the state of protection from infectious diseases. Perhaps the earliest written reference to the phenomenon of immunity can be traced back to the Thucydides, the great historian of the Peloponnesian War. In describing a plague in Athens, he wrote in 430 before Christ that only those who had recovered from the plague could nurse the sick, because they could not contract the disease a second time. Why this is happening? Because they are developing antibodies against those antigens. So that is very important. And the first recorded attempts to induce immunity deliberately were confirmed by the Chinese and Turks in the 15th century. Various reports suggest that the dried crust derived from smallpox pustules were either inhaled into the nostrils or inserted into small cuts in the skin called variolacin. In 1718, Lady Mary Wortley Montagu, the wife of the British ambassador to Constantinople, observed the positive effects of variolation on the native population and had the technique performed on her own children. And after that, there is a very important experiment is conducted by the Edward Jenner in 1798, intrigued by the fact that milkmaids who had contracted the mild disease cowpox were subsequently immune to smallpox, which is a disfiguring and often fatal disease. Jenner reasoned that introducing fluid from a cowpox pustule into people might protect them from smallpox. To test this idea, he inoculated an 8-year-old boy with fluid from a cowpox pustule and later intentionally infected the child with smallpox. Jenner's techniques is of inoculating with cowpox to protect against smallpox spread quickly throughout the Europe. However, for many reasons, including a lack of obvious disease targets and knowledge of their causes, it was nearly 100 years before this technique was applied to other diseases. This is very important. Pasteur extended this finding to other diseases, demonstrating that it was possible to attenuate or weaken a pathogen and administer the attenuated strain as vaccine. In a cow classic experiment as Pouilly Fort in 1881, Pasteur first vaccinated one group of sheep with heat-attenuated anthrax Bacillus anthracis. He has challenged the vaccinated sheep and some unvaccinated sheep with a virulent culture of the bacillus. All the vaccinated sheep lived and all the unvaccinated animals died. This is a very important experiment conducted by the Edward Jenner. Many from some different backgrounds, some are from zoology, some are from botany, some are from biotechnology. So if you are from botany, you are not come across this term, the immune system, because plants are not having the immune system itself. They are having immune system like system in their things. Initially, a different serum component was thought to be responsible for each activity, but during the 1930s, mainly through the efforts of Alvin Cabot, a fraction of serum first called gamma globulin was shown to be responsible for all these activities. The active molecules in the immunoglobulin fraction are called antibodies. In 1883, even before the discovery that a serum component could transfer immunity, Elie Machnikov demonstrated that cells also contribute to the immune state of the animals. He observed that certain white blood corpuscles, which he termed phagocytes, were able to ingest microorganisms and other foreign material. Here we have to understand what is phagocytosis. There are two types of things which cells are definitely doing. One is phagocytosis, that is the ingesting of foreign materials, and pinocytosis. Pinocytosis is cell drinking and phagocytosis is cell eating. This is a very important thing in the endocytosis. In due course, a controversy developed between those who held to the concept of humoral immunity and those who agreed with Machnikov's concept of cell-mediated immunity. This is very important again. So, early theories attempted to explain the specificity of the antigen and antibody interaction. This is very important. We have to understand when an antigen is entering to our body, antigen is having a epitope. Always remember the epitope is present on the antibodies, antigens, and paratopes are present on the antibodies. Okay, so the work of Karl Landsteiner and those who followed him showed that injecting an animal with almost any organic chemical could induce production of antibodies that would bind specifically to the chemical. These studies demonstrated that antibodies have a capacity for an almost unlimited range. Earliest conception of the selective theory dates to Paul Ehrlich in 1900. In an attempt to explain the origin of serum antibody, Ehrlich proposed that cells in the blood expressed a variety of receptors, which he called side chain receptors. The immune system includes innate and adaptive components. So we have to understand here what is the innate immunity. Innate immunity is the things which we are having from the birth, just like the skin provides the first line of defense against infection. Most components of innate immunity are present before the onset of infection and constitute a set of disease resistance mechanisms that are not specific to a particular pathogen but include cellular and molecular components that recognize classes of molecules. And second thing is adaptive immunity does not come into play until there is an antigenic challenge to the organism. Adaptive immunity responds to the challenge with a high degree of specificity as well as the remarkable property of memory. Typically, there is an adaptive immune response against an antigen within 5 or 6 days after the initial exposure of that antigen. So, we will discuss what is innate immunity. Innate immunity is, there are three types of barriers we are having. One is anatomic barriers, physiological barriers, and phagocytic endocytic barriers. What is anatomic barriers? Anatomy means we are having from the birth, that is skin, mechanical. In the skin we are having some pH, that is around acidic pH, so it retards the growth of microbes. Mucous membranes, normal flora compete with microbes for attachment sites and nutrients. Mucus entraps foreign microorganisms. Cilia propel microorganisms out of the body. Some physiological barriers, as we all know, we are having fever. So why we are having fever? When we are having microorganism infestation in our body is rising the temperature by the instruction of the brain, and in the very high temperature there is a microorganism cannot proliferate. So low pH, acidity of stomach contains kill most ingested microorganisms. As you all know, we are having HCl in our stomach, and the pH of that acid is around 3. So chemical mediators, lysozyme clips bacterial cell wall, interferon induces antiviral state in infected cells, complement lyses microorganisms or facilitates phagocytosis. Toll-like receptors recognize microbial molecules, single cell to secrete immunostimulatory cytokines. Collectins disrupt cell wall of the pathogens. Phagocytic or endocytic barriers and inflammatory barriers. So with this we have understood what is the immune system in our body. So in the innate immunity system, what we are having is, the first thing is skin. How the skin is performing its role in immune system. Physical and anatomic barriers that tend to prevent the entry of pathogens are an organism's first line of defense against infection. The skin and the surface of mucous membranes are included in this category because they are effective barriers to the entry of most microorganisms. The skin consists of two distinct layers, as we all know, that is epidermis and the dermis. The epidermis contains several layers of tightly packed epithelial cells. The outer epidermal layer consists of dead cell and is filled with the waterproofin called keratin. The dermis, which is composed of tissue, contains blood vessels, hair follicles, sebaceous glands and sweat glands. So sebum consists of lactic acid and fatty acids, which contain the pH of the skin between 3 and 5. So it will resist the multiplication of microorganisms on the skin. Breaks in the skin resulting from scratches, wounds or abrasion are obvious routes of infection. The skin may also be penetrated by biting insects. If these harbor pathogenic organisms, they can introduce the pathogen into the body as they feed. The protozoan that causes malaria, for example, is deposited in humans by mosquitoes when they take a blood meal. Conjunctiva and the alimentary, respiratory and the urinogenital tracts are linked by mucous membranes, not by the dry protective skin that covers the exterior of the body. So in the other anatomical barriers, the second thing what we are having is the mucous membrane. As you all know, in our nose we are having mucous membrane, in our ear we are having mucous membrane, in our urinogenital system we are having mucous layer. What is this mucous layer? Is in the lower respiratory tract, the mucous membrane is covered by the cilia. Cilia is, what is the ciliary movement is, if something is entering to your nose, generally we are going for the sneezing. What is that sneezing? That is of the ciliary movement. Because of the sneezing, the ciliary movement is downwards, and because of the force of the sneezing, the whatever foreign material is going through our nose to our body, it will be expelled out. Some of the normal flora generally outcompete pathogens for attachment sites on the epithelial cell surface, and in physiological barrier we are having lysozymes, interferons, complement systems. Lysozymes, a variety of soluble factors contribute to innate immunity. Among them, the soluble proteins lysozyme, interferon and complement. Lysozyme, a hydrolytic enzyme found in mucous secretion of and in tears is able to cleave on peptidoglycan layer of the bacterial cell wall. Interferon comprises a group of protein produced by virus infected cells. This is very important. Whenever we are going for some examinations, there it will be asked interferon is secreted by what? Interferon is secreted by the virus infected cells, so that body will know that these cells are virus infected. Complement. So in our next topics we will discuss what is the complement system, what are the lysozymes, what are the interferons. We are going in our next classes, before the situation is cleared, we will go for some other topics on the immunology system and how it will be performed, how it will be done. We will do that. Okay. Thank you very much for listening to me, and God save our world. Thank you.